Hello students, today's topic is sample collection and lab diagnosis of COVID-19. So why it is necessary? Now pandemic is over. Why it is necessary? Why this lecture is necessary? Now virus is adapted to the human population and now there is a tendency to become seasonal. It's like H1N1 and there are comorbid conditions. The government of India guidelines, government of Karnataka, they have given guidelines that we should be cautious for comorbid conditions. And as a clinician, basically, it is necessary to know how to collect the samples. And similar protocol is for other respiratory viruses. Suppose a case of H1N1 or RSV, if you are RSV, if you are suspecting. So this particular how to collect the sample, it will also encompass the sample collection procedures for those viral infection also. So, and in future, such respiratory viruses may more and more may come into the picture. So that's why this information will be helpful not only for COVID-19 but for some other viral infections. And all of you know that if some viral infection is there, there is again propensity to cause the pandemic because rapid spread. Then again, recently, government of Karnataka has given instructions for some increasing the testings. So all these are the points which are necessary to emphasize the need of this particular class. Now, this is the government guidelines. So they have given all SARI cases should be compulsorily tested. Then ANC cases with symptoms. Then pediatric individuals with symptoms or old age individuals above 60 years age with symptoms. They should be compulsorily tested for COVID-19. Presently, as per the guidelines given in the month of May, 23rd May 2025. So these are the designated labs as per the government guidelines and as per the international guidelines. It has been informed that we should be cautious for monitoring the this COVID-19. Then, as per this guideline, government of Karnataka, SARI should be tested compulsorily, and only 5% of ILI. Then people with symptoms having comorbidities like heart disease or chronic kidney disease or some lung disease. Then pregnant women with symptoms, they are being also advised to be tested. Old age individuals above 60 with symptoms also advised to be tested. Then now come to the basic aspects. Little bit I will just cover little. Coronaviruses cause respiratory tract infections like in man, illness which is ranging from mild common cold to severe disease like pneumonia. It is an envelope virus or petal or club-shaped structures, crown-like papillomas are there on the surface. These are the spikes. These are the papillomas giving appearance of solar corona. Therefore, it is called as coronaviruses. It is a large 120 by 160 nanometer in size, spherical viruses, and helical symmetry. So nucleic acid and capsid. Nucleic acid shows helical symmetry. The genome linear and positive sense single stranded RNA about 26 to 36 kilobase pair size and largest among the non-segmented RNA viruses. So these are some basic things about the virus. What is the virus? So you can see this is the electron microscopic structure and this is the diagrammatic representation, imaginary representation. You can see the in the center nucleocapsid, which is showing helical symmetry is there. Then this is the envelope and there is a spike glycoprotein. Then this is in between that purple color is a membrane protein. Then small envelope protein, nucleocapsid protein, RNA. All these are the structures which are there in the morphology. So many coronaviruses, they are causing the disease. So coronavirus causing the mild form of disease, coronavirus 229E, NL63, OC43, HKU1. So these are the coronaviruses produces mild disease. Then coronavirus that could cause explosive outbreaks of severe respiratory disease with higher mortality. Like for example, SARS-CoV virus 1, it has caused explosive epidemic that is called as SARS in China in 2003. Then MERS, Middle East Respiratory Syndrome coronavirus. There again, there was explosive epidemic MERS in Middle East in 2012. Then presently that COVID-19, that is the SARS-CoV-2 severe acute respiratory syndrome coronavirus. The causative agent is ongoing explosive pandemics affecting whole world 1920, and it's also called as COVID-19 or coronavirus disease 2019. So you can see it has been evolving since 2003, SARS, then MERS, and then SARS-CoV-2, that is COVID-19. What are the clinical features? Majority of the infections are asymptomatic. Some people will present with influenza-like illness, and few people they will suffer from severe acute respiratory syndrome. What is the reason of hypoxia? That is because uh, there is a cytokine storm. You can see presence of SARS-CoV-2 infecting alveoli, leading to inflammation, and there is a release of pro-inflammatory cytokines, causing cytokine storm, such as interleukin 2, interleukin 6, interleukin 7, interleukin 8, 10, GCSF, interferon gamma, tumor necrosis factor alpha, and others. So there is a pulmonary edema. So all these factors, accumulation of the inflammatory cells, then neutrophil, all these factors leads to decrease in the oxygen transport, that is leading to the hypoxia, and this is called as cytokine storm. At a time there is a release, and that is the cause of hypoxia, and that is the cause of mortality in those cases. You can see the this there, because of inflammation, the D dimer levels they are released. Then coagulation also, there is a tendency of coagulation, vascular leakages. So these are the further complications. So our purpose was to emphasize the laboratory diagnosis aspects. Now you can see this is in the diagram that there are various modalities of diagnosis which are available. First one, if you go to the first step in any laboratory diagnosis, that is the sample collection. So various samples which are collected, you can collect nasal sample, nasopharyngeal, oropharyngeal sample collection. Then this is a PCR, that is a gold standard for diagnosis of COVID-19. We can go for electron microscopy. These are the microscopic structures, but just it is for this complete the list. But most practical is a RT-PCR. As all of you know that RT-PCR was extensively used for diagnosis of COVID-19 in the pandemic. So rapid tests are also there. Rapid tests, just we have to collect the sample from mid turbinate of the nose to soak the nasal secretions and go for the rapid immunochromatographic test. The test will be available within 20 minutes. So that is also rapid antigen test. What is the drawback? I will tell you afterwards. Then gene expert test, what we are using for diagnosis of tuberculosis, same machine can be used by using different kits. Now you can see expert SARS-CoV-2. So these are very costly, like 2,500 rupees cost. So such buckets will be available, such cassettes will be available. We have to take the patient sample secretions and inoculate that, and it should be run. So again, the principle is PCR only, RT-PCR. It is entirely closed system, so that machine can also be used for doing the RT-PCR. But however, the only at a time just 4 slots will be there for one machine, and we have to run for 2 hours for each once we load entire machine. So that much output will not be there. So it is more time consuming, costly. Now, always it is very important as a doctor, clinician always use PP before collection of the sample. So whenever any respiratory viral infection occurs in future also, always personal protective equipment is to be done. Donning and doffing, as all of you know, that it should be follow the particular sequence should be followed. So we have to go from down to up for donning, and for doffing up to down. So all those precautions. Mask should be removed in the end. Mask should be removed in the end. So during collection and the pandemic, they have prepared such kiosks. So we can collect. The technician is collecting the specimen from the lady who came for the diagnosis. So that can be helpful because each and every time changing the PP, so it will save that. So kiosk can also be used. It is available in our OPD. So those cases OPD cases can be taken for the samples can be taken for through the gas. So indeed necessary things. Those doctors who are working in ward, they should get trained themselves. Already we have trained some doctors, so they should get trained themselves for collection of sample. So the necessary things should be indented like viral transport medium. It will be available in microbiology laboratory. Sometimes depending upon the swabs. Swabs can be broken. There will be some weak thin point where it can be broken. But if such swabs are not available, we have to use scissors. Then ask the lab for VTM. They instruct the patient. Patient relative should go, or patient or nursing orderly, or staff of the hospital should go and prepare the specimen referral form. For specimen referral form, the mobile number of patient is important because OTP will come and it will get the patient will also receive the report once it is entered in the ICMR portal. Then here before that, referring doctor should fill the form and it should submit to the lab, and lab technicians, two technicians are there in the lab. Their mobile number is mapped with ICMR portal, so they will generate the SRF and state what is the indication, because presently I told you SARI, then old individuals with comorbidities, then ANC cases and pediatric populations. The present indication in May, June 2025, these are the indications. You can see the photograph here, VTM is there, swabs are there, you can see. And sometimes if the swab is cannot be break, then scissors may be required because the length of swab will be more. So we have to cut it so that it will be adjusted in the VTM tube. The samples should be immediately transported to the microbiology laboratory and there should not be delay. In case there is a delay in transfer of the sample, transportation of the sample, it should be kept in the refrigerator. Cold chain should be maintained. So the doctor should instruct the lab and give necessary details to generate SRF by our technician. That already I told you. So this is the website where any other person cannot do. Only the number should be registered with this, then and then only it will show. And so it will be registered as a sample collector. So you can see how this comes. RT-PCR test of India, you can see name, then mobile number. Mobile number is important. Mobile number of patient is important. Get OTP, then patient will give the OTP, it should be fed. So that it is ensure that we are given correct number and the report will also be dispatched immediately on the mobile. So while collecting the sample, ask the patient to tilt 70-degree angle backside and we can collect the samples. Anterior nails rarely, but mid-terminate. For rapid antigen kit, the samples should be collected from mid-terminate. And then for RT-PCR test for COVID-19, nasopharyngeal and oropharyngeal swabs are added in the same VTM. Both the swabs, they are put in the same VTM and immediately transported. If there is a delay in transport, I repeat that we should keep in the cold temperature, 4 degrees Celsius. So you can see the photograph. A little bit tilting of the head is necessary to visualize. And for suppose if you are taking oropharyngeal sample, tongue depressor is necessary because tongue will come in between. So tongue depressor is necessary as a additional requirement of the for sample collection. There may be gag reflex or patient may feel uncomfortable during collection, even nasopharyngeal also, because we are penetrating 5 to 6 centimeter inside the nose. So we have to pass horizontally. So you have to pass horizontally. You can see here, so that it will reach the nasopharynx. So little bit discomfort is there. So before collecting the specimen, we should inform the patient that such thing will happen. So patient's mind will be prepared for that. That's after collection, oropharyngeal and nasopharyngeal specimen, it should be submitted to the laboratory. Don't forget to generate SRF. Again, I am repeating this. So on the ICMR website, it is as per the government guidelines. It will be done by our lab. If there is a delay, 4 degrees Celsius temperature preservation is important. Otherwise, the attempt will be futile. So throat swab, nasal swabs. So you can go for NAT, nucleic acid amplification testing, real-time RT-PCR, then automated format CBNAT and TrueNAT. Already I shown that machine. So these are the gene target E, N, and M genes, confirmatory RDRP gene, N2 gene. So there will be two genes which are amplified. Any one of these E, N, M. So screening genes. Then confirmatory, that again RDRP or N2 genes, that will be confirmatory. So simultaneously we are amplifying the gene targets. So this gene target, one is a screening gene, another one is a confirmatory. Both should, there should be amplification of both. Along with that, there is a internal control showing that human tissue is there. So you can see here, though this graph will be obtained on the RT-PCR machine. On that LCD display, you can see there is a increase in the number of copies. So real time, which will be shown on the in the form of graph. If it is negative, so there will not be any formation of graph. So you can give copying cycles and machine will give cycle threshold. So sometimes 24 or 20 cycle threshold means it is highly positive. If cycle threshold is 45, that means 45 cycles required for achieving the detectable level of gene. So that means it's having more cycle threshold. So viral load is less. And each kit will give some interpretation criteria, what should be considered as CT value. And depending upon that, report is given. And all this report can be documented. It can be stored in the computer for further reference in case if there is some discrepancy or there is some problem, we can refer. So that data can be saved in the computer. Then antigen detection assay, that is called as RAT, rapid antigen test, detect the nucleocapsid protein antigen in nasopharyngeal swab. Antibody is not useful for the acute case. Antibody detection is used for only serological surveillance or survey in high risk or vulnerable group, and not for clinical diagnosis, because that will be acutely presenting antibodies. For development of antibodies, it will take much more time. So it will not be helpful. So you can see RAT procedure. It's very simple. We can take from the mid terminate, penetrate about 3 centimeter inside, ask the patient to tilt the head like 70-degree angle, and with the help of swab, we have to keep it for about 3 to 4 seconds inside, so that nasal secretions which are there, it will get soaked inside the swab. And there is a cuvette given. It will be closed. We have to open it and put the swab. So all the antigen will get dissolved in that. It will get disrupted, and this solution, one drop, 4 drops are added in this slot. And slowly we have to see by immunochromatographic method, there will be percolation, there will be spread of the capillary action, spread of the that solution in capillary action. It will show the test as well as control band. If only control comes, test is there is no band, that means negative. If both band comes, it is positive. But what is the problem with rat? If it is positive, as per the ICMR guidelines, it is a confirmed positive. I repeat, if it is positive, as per ICMR guidelines, it should be considered confirmed positive. If it is negative, it will not rule out. So, if it is negative, we have to collect the sample for RT-PCR. So, what is the guideline given by ICMR? Simultaneously, you collect both specimens. That is rapid antigen test as well as RT-PCR specimens should be collected simultaneously. If RAT is positive, that discard that RT-PCR sample, because positive is compulsorily it is positive, confirm positive. If it is negative, we have to process the RT-PCR. So simultaneously both the samples are taken for RAT as well as RT-PCR. So what is the plan if a rat comes negative? Drawback of rat, already I told you. The negative test result will not rule out the COVID-19. So negative will not rule out. So therefore, in those cases who are symptomatic and fitting in the criteria of clinical criteria, they should be tested for RT-PCR. Is SRF generation necessary for rat? Yes, SRF generation is also necessary for rat. And for rat test, if it is negative, there should be data that it is sent for RT-PCR. So all these are the ICMR guidelines. So there will be certain situations where some of the percentage of the specimen should be sent for sequencing. So there will be sequencing setup will be very costly. It will be sent. It will be sent in Apex labs where the totally dedicated for virology work, and it should be the sequencing should be done in those labs. And gene sequencing setup requires a lot of infrastructure and equipments which are costly. So viral genome. So what is the present in the May and June 2025? What is the scenario? I will tell you which is the sequence, which is the subvariant presently. Then viral culture, only research purpose. Viral culture is also used for vaccine preparation. All of you know Covishield, Covaxin, all those. That viral culture, for that purpose, viral culture is required, but viral culture is not required for diagnostic purpose. Then non-specific tests, as all of you know, clinically, radiologically, you can go for chest X-ray, where ground glass appearance is seen. Then biomarkers, interleukin 6, D dimer, So that can be used for detection, and we can detect the biomarkers. And but it will be, these are the indirect tests. So now this is the last line. The Indian strains have reported surge in the cases. Actually, in May they have given. Well, before that, it started Maharashtra and all those places, Kerala, all the states they started reporting, and Karnataka on 23rd May they have given the notification for implementation and increasing the testing. So surge. So I told you after sequencing, it has been found that Indian states have reported a surge in cases reportedly linked to the newly identified subvariants NV 1.8.1 and LF 0.7 point. So these are the strains. And how it is detected? It is detected by gene sequencing. So these variants have undergone mutations and show genetic difference compared to the earlier strains. Now, because of these mutations, now this strain is highly transmissible, but mortality is less. But however, we should be cautious for those with those comorbidities, comorbid individuals, then ANC cases with symptoms, and those cases, pediatric population with symptoms, as per the government guidelines. So this is just a video for this lecture on the lecture of sample collection and brief, and mostly highlighting the practical aspects of this. Thank you very much for your patient listening.